So, question number 7. Only monosaccharides and most disaccharides are reducing sugars. Reducing sugars produce a red precipitate when heated with blue Benedict's reagent. Identify the row that shows the correct results after heating 3 carbohydrates, maltose, amylose, and fructose, with Benedict's reagent. Right. So you would have come across this in class, the Benedict's reagent and the types of carbohydrates, because it is part of the specification. We would have also done a lab experiment involving this type of reaction where you would have tested different carbohydrates with Benedict's reagent to see which ones form the precipitate. And so you would have also learned an important phrase, and that's every sugar is a reducing sugar except sucrose. And so that says that whenever you put sucrose with Benedict's reagent, it will show no precipitate. However, other sugars will. And other reducing sugars are commonly monosaccharides and disaccharides, as stated here. And those are your glucose, your galactose, your lactose, your maltose, your fructose. And so if we come onto here, we can see it's maltose is the first option, and maltose is made of 2 glucose units, and so it will show a precipitate. Amylose, is a polysaccharide, and although it's composed of many glucose units, it's a complex carbohydrate, and so you don't have polysaccharides interacting with Benedict's reagent. And if you didn't know this, it gave you a clue in the statements above. So only monosaccharides and most disaccharides are reducing sugars, and so that means polysaccharides are not. And you know that amylose is a polysaccharide, so that would have given you an automatic hint that it wasn't going to be maltose because it's a polysaccharide. Fructose is a monosaccharide, and so of course it will show a red precipitate, and so the answer is B. The Benedict's test can be made quantitative and used to determine the concentration of a glucose solution. The red precipitate formed is removed by filtration. The precipitate is dried and the mass is recorded. The table shows the mass of precipitate formed from a range of glucose concentrations heated with 5 centimeters cubed of Benedict's reagent. Right, so at first glance we can see that as we're increasing the concentration of glucose, the more precipitate is being formed. However, we can see over here that as soon as we reach 16 milligrams per centimeters cubed, the mass of precipitate stays the same. So, analyze the data to explain the relationship between the glucose concentration and the mass of precipitate formed. Okay, so whenever you get an analyze and explain data, what that basically means is state what you see and tell me why that happens. And so because it's 4 marks, you're going to have to give 2 points that you see here, and you're going to have to explain why those 2 points occur. And so when we had a glance, you could automatically see that when we increase the concentration, we're going to increase the precipitate, point number 1, and until we reach 16, and the mass formed is going to be the same. And so those are your 2 points. But now we have to explain them. And so it's nothing that you would have not learnt in class. So let's start answering. So we said that as you increase the glucose concentration, the mass of precipitate increases, and this is true from 0 to 12 milligrams per centimeter cubed. So we say concentration increases, mass of precipitate also increases. Okay, so now we need to say why. And you know that, well, the glucose is going to interact with the Benedict's reagent, and that's what's forming the precipitate. And so that's your answer there. And that's because all the glucose reacts with the Benedict's reagent. reagent. Okay, so that's our first 2 marks. Now we need to analyse the other point. And the other point is basically the mass of precipitate stops increasing above 16 milligrams per centimeters cubed. So the mass of precipitate stops increasing after 16 milligrams per centimeters cubed. And now we need to say why. And that's because all the Benedict's reagent is being used up and the glucose is the excess, because we're increasing the mass of glucose, but the volume of Benedict's reagent is the same. So there will come a point where you've got too much glucose molecules and the Benedict's reagent is all used up. And so you would actually have a graph that looks like this. And that's what's happening here at 16 and above, is that we have no more Benedict's reagent. So, because all of Benedict's reagent used up, and your glucose is the excess. When the investigation was repeated, the mean error for each measurement was calculated. The mean error for each measurement was 0.05 grams. Calculate the percentage error for the mass of precipitate measured at the glucose concentration of 2 milligrams per centimeters cubed. Right, so you would have come across percentage error if you study chemistry, because these are frequent questions that come up and you would have done calculations for them. In biology, you would have probably done them once if they came up in a practical write-up, and it's nothing as difficult. It basically means that the percentage error is the difference between the experimental value and your value, so the actual value, the value that you've obtained. And so because it's a percentage, we're going to be timesing by 100 at the end. Whenever there's a percentage, to get a percentage, you times by 100. So your final answer is times by 100. And to get the percentage error, we need the experimental value and we divide it by the actual value obtained. And so they always give you the experimental value and they're telling you that it's 0.05 grams. So straight up, that's our experimental value. And we're going to divide it by the actual value, or the obtained value that we got. So if someone hasn't come across this type of question before, they're probably going to have a slight panic and go, okay, I'm dividing it by this number. But actually, if you look carefully, you're going to see that it says calculate the percentage error for the mass of the precipitate measured at the glucose concentration of 2 milligrams per centimeter cubed. So they want the mass of the glucose that's measured at this point. And so if we scroll up, the glucose concentration, they wanted 2, so we go to 2, and we see the mass that's formed is 0.28 grams, and that's going to be our bottom number, 0.28 grams. And so your calculators are allowed, and you times it by 100 to get it as a percentage, and the answer would have been 17.85, and that's just abbreviated to 17.9%. That's your answer. Always just write it over here, because sometimes some examiners can be a bit fussy and they always just want the answer over here. So if you've written it over here, don't forget to just add it here. But that was 1 mark. Typically, these type of questions can be 2 marks. You get 1 mark for this and 1 mark for the answer. But here they've only given you 1 mark. Explain one way in which the mean error in measuring the mass of precipitate could be reduced. So once again, this type of question you wouldn't really study it in a biology textbook. You just study the specification and what you need to know for the exam. And sort of these questions don't really come up unless you've done the practical and you've understood every aspect of the practical and you know how to reduce errors. And once again, if you study A-level chemistry, this answer would have been easier for you. But it's basically asking you, how would you reduce errors from occurring in this experiment. And they will explain one way, and that basically means you have to give one example and explain how that will reduce the error. And so for these type of questions, you can always sort of memorize the answers, because if they bring another question in a different exam paper, it's going to be the same answer. And so you can always sort of like learn what type of answers they're looking for in these questions, because it's always going to be the same answers. And so you have these sort of, we'll go through it now, the types of examples you can give. And so a good tip is to just learn the examples, have them in the back of your mind. So when you get a similar question like this and they tell you how do you reduce errors, you will know how to answer it. It's particularly helpful in A2 or Year 13 or A level in paper 3, when it's heavily sort of like practical based papers, when they're looking for, how would you do this experiment correctly, or, sort of questions about how would you reduce the error. So it's good to have, like, you've come across this question now and to have the answer in the back of your mind. So we'll go through different examples, the ones that you could learn. So first one, the most commonly used one and the easiest one, is you reduce errors by repeating the measurements. So you do repeat measurements. And you get 1 mark for that. And now you need to say, well, how does this reduce the errors? And here they've written because it reduces standard deviation. So reduces the standard deviation. Now, you would have come across standard deviation if you studied A-level psychology. You would also come across standard deviation after doing sort of the field projects, where you would where you would come across different types of statistical tests, and you would come across this type of wording, standard deviation. And standard deviation just means how much does your result deviate, so move away from the normal value or the standard value. And so the more that you repeat experiments, the more you can sort of get a mean value. And so it reduces standard deviation because you have less results that are likely to be sort of anomalies or results that are far further away from the normal. And so another way you can also, in other exam papers, you can get away with sometimes, if you forget standard deviations, you can say repeat measurements because they increase reliability and validity. So it's worthwhile just to remember that whenever you write repeat measurements, reduce standard deviation, you can also say it increases reliability and validity, because I have actually seen in papers you do get marks for saying reliability. Although here they haven't stated it, but in other papers they do. Right, what's another example is that increased drying time. Whenever you have a precipitate forming, and they've told you, if we go back to here, and they've said the steps, whenever you have a question that's telling you, well, how would you reduce errors, go back to the method and start picking out different things you can improve. Whenever they say something has to be dried, that's automatically you get a mark for saying increased drying time. And that's because the more you leave something to dry, you ensure all the water is being removed. And so therefore, when you start to measure the precipitate, you know that you're actually measuring the precipitate instead of other water molecules still trapped. And so that's another point, is increased drying time. And that's because it ensures all the water is removed. Okay, another point you could have mentioned is whenever you're also measuring something and you're measuring the mass, you're going to be using a balance. And so always say, check the calibration of the balance. Whenever you hear the word calibration, it just means standardize. And so, because whenever you calibrate something and you standardize it, you increase the accuracy of the measurement. And so, increases accuracy of the measurement. Right. Another one you can say is whenever you're using filtration, you're going to be using a filter paper. And so it's worthwhile in biology saying to rinse off all the precipitate from the filter paper. From the filter paper. And that's because it ensures all the precipitate is removed and measured, so all the precipitate, sometimes that's an abbreviation for precipitate, you might see PPT. So all precipitate is collected and measured. And so it's worthwhile just to remember these. So whenever you come across a practical, you can just, and how to reduce the error or how to improve the accuracy of the practical, you can just list one of these. And that'll be it.